Yeah, so for today's Data for the People session, we're going to be discussing a paper that goes into the intersections of COVID-19 and type 2 diabetes. So just before I start talking about the paper, I just want to go into a quick outline of what I'll be discussing throughout this talk. So first, I'll talk a bit about what exactly we're discussing and why this is relevant to us and exactly why the authors decided to research this specific topic. Second, I'll be going into some general definitions of what exactly diabetes is, and then what a comorbidity is, and then what a significance test is, which is something that's used really throughout the study. Next, I'll be talking about how exactly the authors approach this issue, so what techniques they utilize. And then I'll talk a bit about what exactly the authors add to the field by a discussion of the data that the authors present in this paper. Lastly, I'll discuss some conclusions made by the authors, and in this discussion, I'll also talk about the clinical significance of the authors' findings, as well as some limitations in future studies of this research. Okay, but before I go into all that, I want to talk a bit about myself. So I was born in Moscow, Russia, and I moved to the United States when I was quite little. I moved to Atlanta, Georgia, and have lived in the United States ever since. So this is me. I graduated from American University. I got my undergraduate degree there in physics and mathematics. And I'm currently attending Weill Cornell Medicine, where I'm studying biophysics for a PhD. And recently, I've really been into just exploring New York City. So this is me right in front of the New York City skyline, and this is on the East River in Williamsburg. And I've also really been into vegan cooking. So here's kind of a blueberry loaf that's vegan that I made recently. And also this is like a really cool vegan Korean dish that I prepared recently. Okay, so first let's talk a bit about what this paper is about and why it's important. So this is the paper that we'll be discussing throughout the talk. The title is The Association of Blood Glucose Control and Outcomes in Patients with COVID-19 and Pre-Existing Type 2 Diabetes. So this paper is what is known as a clinical and translational report, which just means that it is typically done on human participants, and it's more so directed to direct health outcomes in the present. And lastly, this paper is published in a journal called Cell Metabolism, and that means that this paper is fully peer-reviewed, and that just means that it has gone through a process where other scientists in the field have gone over this paper and have decided that it is rigorous enough to be published as a scientific article, excuse me. Okay, so now I kind of want to go more so into what clinical and translational research is as compared to what we call basic science research. So first, basic science research is more fundamental and foundational research that typically is designed to kind of pave the way for more applied science and medicine. And this typically involves reducing particular problems to really their simplest terms, and then using model systems in order to tackle those problems. So think of using different kinds of cell types and animal models. So you can think of using like lab mice for different kinds of experiments. And then sometimes computer simulations are also used. And then clinical and translational research is more so research that takes from basic science research and then applies that to medical projects specifically. So it's more so designed to impact health outcomes directly. And in the case of clinical research, it typically involves human participants. And the kind of research that we're going to be discussing today is clinical and translational. So these two being both like really rigorous kinds of research are very similar in many aspects. So both kind of utilize very similar scientific research and principles, as well as pretty much the same kinds of like research methodologies. And they both involve like really rigorous collaboration between different kinds of specialists, especially in the field of medicine. Both kinds of research really incorporate specialists from both fields. And ultimately, they share the same goal, which is the advancement of knowledge, and in the field of medicine, of course, the improvement of health outcomes. Okay, so going back to the paper a bit, I kind of just want to dissect this title. So again, it's the association of blood glucose control and outcomes in patients with COVID-19 and pre-existing type 2 diabetes. So what this title is basically saying is that it's presenting research about the outcomes in patients that have type 2 diabetes that contract COVID-19. And this word association just means that the authors are attempting to compare two groups and find some sort of correlation between them. In this case, those things being COVID-19 and type 2 diabetes. But when scientists want to establish correlations, the style of scientific reasoning that they're typically using is called probabilistic reasoning. And this is just the use of statistical methods in order to establish regularities, as well as identify patterns within data trends, and then establish significance in those trends. So when scientists use probabilistic reasoning, again, they're using statistical analyses for particular data sets that they have in order to establish significance in those data trends in a rigorous way. Okay, so now I'm just going to discuss a bit about why this research is really important and why these researchers published this study. So first, diabetes is really a major non-communicable disease worldwide. And actually, many scientists have called it a pandemic of itself. So kind of understanding diabetes in the context of COVID-19 is really important given its really widespread nature and how quickly it's been spreading throughout the world. Second, so diabetes is known to present a really steep decline actually in survivability when it's combined with respiratory illnesses. So it's already been known that diabetes is kind of a really major pre-existing condition for a lot of diseases, especially respiratory illnesses. And actually, a really similar study to this one had already been done with diabetes and the SARS epidemic, which took place in 2003 in East Asia as well as throughout the world. So the authors of this paper really want to show the same result rigorously for COVID-19. And lastly, diabetes is known to affect certain populations really disproportionately, as was previously mentioned. And this really represents a really serious public health and social challenge when we consider COVID-19, just because of its extremely widespread nature and how quickly it spreads, and also the fact that COVID-19 itself also affects certain populations really disproportionately. So studying the interactions of these two diseases is really important for improving the health outcomes of these communities where these diseases have such a disproportionate effect. Okay, so now I'm going to go into some definitions. So I'll kind of define what diabetes is and then talk about what comorbidities are. And then lastly, I'll go into significance testing, which is a technique that is used all throughout this study. And here are just some keywords to keep track of while I'm talking about some definitions. So the first is glucose, second is comorbidity, and then the third is a significance test, as well as a p-value, which is a part of significance testing. Okay, so what is diabetes? So a really general definition of diabetes is that it is a metabolic disorder where the body's ability to process glucose is disrupted. And I've kind of highlighted these two really key terms here, metabolic and glucose. And what metabolic means is it pretty much refers to any process that is kind of biochemical in the body, which serves to sustain life in the body. And then glucose is just a small sugar molecule, which gives energy to your cells. So kind of using those two definitions, diabetes is a disorder where the life-sustaining processes that relates to glucose processing in your body are somehow disrupted. Okay, so now to better illustrate diabetes, I'm just going to talk about what metabolism kind of looks like in someone that is a non-diabetic. So here's me. So let's say that one day I decide to eat a pizza. So as I eat this pizza, it'll travel down my body into my digestive system, and it'll be broken down into several different components. Some of which are carbohydrates, proteins, fats, and several other nutrients, which which I need to sustain my metabolism. And so the one of these that's really the most relevant to our discussion today are carbohydrates, which you can really think of as molecules which kind of give your body sort of like instant energy after you eat food. So once this pizza is broken down into these carbohydrates, the way that your body can utilize the energy from these carbs is by breaking them further down into the glucose molecules that I talked about before. And again, glucose is just a small sugar molecule that your body can use for energy. Okay, so once this process happens, the glucose kind of floats around throughout your body in your blood, and it's just kind of waiting to be absorbed by your cells so that they can use them for energy. But the problem is glucose actually can't be directly absorbed by cells. The glucose can't penetrate the cell right away. So what the body does is it utilizes a specialized organ called the pancreas. And what the pancreas does is it can basically sense when there's a lot of glucose in the body after you just ate a meal, for example. And then it releases a hormone called insulin. So once insulin is released into your body, it attaches to your cells, and what happens is it acts kind of like a key to like a locked door. And then once this happens, the glucose can then absorb into the cell and give it energy. So what happens in the case of a person that has diabetes? So one of two things, or actually both, can happen in the case of diabetes. The first is called insulin resistance. And essentially what this is even when insulin is released into your body and attaches to your cells, the glucose that's floating in your blood, it still can enter into your cell. And then what happens is your cells basically can't get energy from glucose, and then the glucose starts to accumulate in your blood. The second thing is insulin misproduction. And this basically happens as a result of some damage that comes from somewhere to the pancreas, and then it's basically unable to produce enough insulin basically to meet the demands of glucose absorption for your body. Okay, so when a person has diabetes and they develop insulin resistance and insulin misproduction, what can happen is since your cells basically aren't absorbing the glucose that you get from eating in your body, the glucose starts to accumulate in your blood, and this can really cause a lot of damage to blood vessels. Then, again, since the cells aren't absorbing enough glucose, they may not have enough energy to kind of do the typical processes that they would typically be doing. And over time, this can cause damage to a lot of organs throughout your body, and eventually cause disease to those organs. And the most common as a result of your cells not absorbing enough glucose are kidney, liver, and heart disease. And all of these issues, given enough time, can lead to nerve degradation, excuse me, which it itself can cause a plethora of issues given enough time. Okay. So the kind of last thing I want to mention about diabetes is that there are two main types. So the first type, usually called type 1 diabetes, is one that typically comes about as a result of genetics. And it's typically a result of some issue in the cells of the pancreas, which produce insulin. And this type of diabetes accounts for approximately 5 to 10% of all diabetes cases. Okay. The second type of diabetes is usually called type 2 diabetes. And this type of diabetes can really result from a plethora of causes. Poor diet can be a factor. Damage to the pancreas can be a cause, as well as genetics as well. And this diabetes, type of diabetes, is commonly called adult onset diabetes because it usually manifests in adults. And this accounts for approximately 90 to 95% of all diabetes cases, and is the type of diabetes that we'll be discussing for the rest of this talk. Okay, so now I really want to define what a comorbidity is, and this is basically just any condition whose symptoms can overlap and potentially exacerbate the symptoms of another condition. So a pretty easy example to think about when thinking about comorbidities is maybe imagine someone that's suffering from asthma. So they have symptoms which may cause them to have trouble breathing. And then let's say that they contract a respiratory illness like the cold or the flu. So the symptoms of that respiratory illness could potentially exacerbate the pre-existing asthma that this person has. And as you can imagine, breathing can become much more difficult for them. So we would say that asthma is then a comorbidity for that respiratory illness. So here I'm just kind of illustrating potential symptoms that could cause comorbidities for type 2 diabetes as well as COVID-19. So in the middle of the Venn diagram between the two conditions, I've listed immune disruption, vascular damage, as well as multiple organ injury. And these are kind of the most severe outcomes of both. But as you can imagine, the most severe symptoms of conditions could lead to the most severe exacerbations in terms of comorbidities. Okay, so now I kind of want to pivot a bit and talk about statistical significance and significance testing, which are two things that are, which are two techniques that are used throughout this study. So first, statistical significance is basically a mathematical measurement of the regularity of some observed effect. And then significance testing are basically any formal methods that are used to determine statistical significance. So just a quick example to kind of illustrate these two concepts. Let's say that we're developing some medicine and we want to test if this medicine has the desired effect that we think it does. So what we do is we give this medicine to one group, labeled group 1 here, and then we give a placebo, which we know definitely does not have an effect, to another group, which is group 2 here. And what we want to do is do an experiment where we basically compare the effects of the medicine on group 1 to group 2, which we know the placebo isn't having any effect on them. So we do this experiment, and then we collect the data, and then we put it through a significance test. And what a significance test really is just a mathematical formulation, typically, where the input is the data that you get from an experiment. Okay, and what the significance test gives you is what is called a p-value. And what you can really think of a p-value is basically it's a probability that describes, or it's the probability that the outcome that you got from your experiment came about as just pure chance. So in this case, from the experiment, let's say that we got that the medicine actually did have like some sort of effect on group 1 as compared to group 2. So this corresponding p-value we got, which is 0.045 or 4.5%, is basically saying that the probability of the outcome that we got, which is the medicine actually did have an effect on group 1, just coming from pure chance, is very low, or approximately 0.045 or 4.5%. And so given this p-value, we can say that we're pretty sure that the results that we got from this experiment is significant, and then we can say that we probably think that the medicine actually definitely does have a significant effect. Okay. So just a quick poll about p-values to kind of drive this concept home. So the poll states, given that a p-value represents the probability that a phenomenon happens by pure chance, what p-value would we expect to get from a significance test of something that most probably is not a random occurrence? And the answer choices are A, 0.9 or 90%, B, 0.001 or 0.1%, C, 1 or 100%, and finally D, 0.67% or 67%. Excuse me. All right, so looks like most people said B, which is the correct answer. And remember, this is correct because the p-value represents the probability that a phenomenon happens by pure chance. So if we're testing something that, that we know is definitely not a random occurrence, we'd want the p-value to be extremely low because that would tell us that our intuition about that is correct. Okay. So going back to the keywords, so yeah, just keep these keywords in mind as we move along in our discussion. So number one is glucose, which remember is the small sugar molecule, which gives your cells energy. And then comorbidity is any condition whose symptoms can overlap with another's to exacerbate them. And then lastly, significance test, which is basically any formal test of significance, as well as the p-value, which is the probability that the outcome that you got came about as came from pure chance. Okay. So now I'm going to go into the methodology that the authors use in this paper. Excuse me. So how do the authors approach this issue? So before I get into the exact methodology, I just want to mention that this is what is called a retrospective, longitudinal, and multi-centered study. So what do these words mean? So a retrospective study is any one that compares two groups of people where the groups differ by one or more factors of interest and where the data is collected prior to the analysis. And then a longitudinal study is one that it's one where the same observation basically is made repeatedly over a set period of time. And then a multi-centered study is just one that takes place in multiple settings. So keep these words in mind, and then I will come back to them as I talk about the methodology of the paper. Okay, so this figure here kind of presents exactly how the researchers started their project. So let's start at the top of this flow chart up here. So it says that 9,663 patients were diagnosed with COVID-19 and were admitted to 19 hospitals in the Hubei province in China from December 30th to December 20th. So this is the initial cohort or group of people that the researchers started with. And what they wanted to do with this number of people is they wanted to split this group into two groups. One being one with patients that had pre-existing type 2 diabetes, and then another one that did not. So in order to get to these two groups, the researchers took 2 or 3 steps where they essentially eliminated patients who were ineligible for the study. So for example, they eliminated 872 patients that did not have complete medical records and therefore could not participate in the study. Another example, about 1,000 patients who were under the age of 18 or above 75 were eliminated. And then finally, after this elimination process, the researchers got to this final cohort number of 7,337 total participants. Then they split this large group up into two smaller groups, one that had 952 participants, and all of these patients had pre-existing type 2 diabetes. And then at the other, which had 6,385 participants, and this was the non-type 2 diabetes group. And by the way, type 2 diabetes is abbreviated here as T2D. Okay, so once the group had these two cohorts, what they did was they collected several variables for all of the patients. So first, they collected each participant's age and gender. Second, they collected each participant's symptoms upon admission as well as over time. And then next, they collected the comorbidities that were present at admission other than type 2 diabetes. And then they also collected the blood profiles of every patient upon admission as well as over time. And blood profiles are basically kind of like your blood work. So they were just looking for things in the patient's blood which were relevant to the study. So for example, they were looking for glucose concentrations or insulin concentrations, stuff like that. And lastly, they collected the overall management of each patient while admitted. And what management means is basically any medical intervention that any patient had to undergo. So that could have been things like administration of certain medicines to the patients, or the use of some medical instrument like a ventilator. And by the way, just to go back to the previous three definitions, this study is, first of all, retrospective because we're comparing two groups here that differ by some variable that we're interested in. In this case, it's type 2 diabetes. And then it's longitudinal because we're recording all of these variables over and over again, basically over a certain period of time. And then it's multi-centered because it took place in 19 different hospitals where the patients were admitted. Okay, so going further, the researchers kind of delineated the 952 type 2 diabetes patients further into two groups. So the first group was, it was labeled the well-controlled BG group, which BG stands for blood glucose. And in this group were patients that had glucose levels in their blood, which were considered to be in a healthy range. And here, this is considered to be from 3.9 to 10 millimolar per liter glucose in the blood. And millimolar per liter is kind of just a measure of concentration. And the other group was the poorly controlled blood glucose group. And this group had patients who had what is considered to be unhealthy levels of glucose in their blood. And in this case, that's considered to be above 10 millimolar per liter of glucose in their blood. And then once they had these two groups, they recorded all of the same variables that I had just talked about in the same slide for both groups. Okay, so once the researchers had all of these variables, what they did was they compared all of the groups that they had delineated to each other on the basis of each variable. So in this case, the researchers are comparing the type 2 diabetes cohort to the type 2 diabetes cohort on the basis of medical management. And in this case, it's the use of particular drugs. And they recorded, in this case, they recorded the frequency with which each group had to be administered these drugs. So once they had all this data, what they did was they applied a significance test to it, and then they generated p-values for each instance of variable. And just looking at a more specific example, we're going to look at a drug called systemic corticosteroids, which is basically a drug that helps with really severe immune responses. So looking at the type 2 diabetes cohort, we can see that these patients were prescribed this drug at a rate of about 29.4%, compared to the patients in the non-type 2 diabetes cohort, which were prescribed this drug at a rate of about 23%. Okay, So we can clearly see that the type 2 diabetes patients were prescribed this drug at a much higher rate than were the non-C2D patients. So now we want to establish significance in this data, and we want to be able to say that this isn't happening just by pure chance. So what the researchers are doing is they're taking this data and they're putting it into a significance test. And in this case, they get that the p-value for this particular case is actually below 0.001 or below 0.1%. And remember, a p-value is the probability that your outcome came as a result of just pure chance. So in this case, that probability is extremely low, which means that we can say that in fact this outcome is significant and didn't come about as a result of pure chance, and therefore we can say that type 2 diabetes, having type 2 diabetes and COVID-19 is correlated with a higher rate of being prescribed systemic corticosteroids to deal with particular symptoms. Okay, so now we're going to go into kind of the results section and what the authors add to this field. Okay, so the first major result of the study is that type 2 diabetes is in fact a major comorbidity for COVID-19. And here I've presented a table where they present the outcomes or the most severe symptoms that the patients faced. So in this table, what they're doing is they're comparing the type 2 diabetes cohort to the non-type 2 diabetes cohort on the basis of outcome. So from this table, we can see that for pretty much every severe kind of outcome, the type 2 diabetes cohort are experiencing these outcomes at a much higher rate. So if we look at one specific example, which is ARDS, which stands for acute respiratory distress syndrome, which is basically a really severe respiratory outcome, we can see that the type 2 diabetes patients suffered from this outcome at a rate of almost 17% compared to the non-type 2 diabetes patients, which suffered this outcome at a rate of just 7%. And this result, when applied to a significance test, had a p-value of below 0.1%, which again means that we can correlate having type 2 diabetes with a higher rate of ARDS. And this is true pretty much for all of these outcomes, except this one, DIC, which stands for disseminated intravascular coagulation. And this had a p-value of above 0.05. So we can't really say that this result is really significant, but we can't really say that it's not significant from the significance test that was done. And this could probably be due to just like the low number of people who actually suffered from this outcome. But in general, for the rest of the outcomes, the results were extremely significant, below 0.1%. Okay, so the second major result from this study was that control of glucose in the blood is essential to good outcomes in COVID-19 patients with pre-existing type 2 diabetes. And the first kind of chart that I'm going to present for this results is a similar one to the previous one. So in this case, they are comparing the well-controlled blood glucose cohort to the poorly controlled blood glucose cohort on the basis of medication prescription for particular outcomes that they had. So again, for pretty much every single medical intervention that's shown in this chart, we can see that the poorly controlled blood glucose patients had to be administered all of these interventions at a much higher rate. So again, let's look at the systemic corticosteroids. So looking at the poorly controlled blood glucose group, we can see that they were prescribed this at a rate of about 35% compared to 20% for the well-controlled patients. And this result had a significant p-value of less than 0.1%, which means that this result was extremely significant. And this is true pretty much for all of these drugs. Some of them don't have the most significant p-value. For example, this one has a p-value of 0.5%, which means that the probability of this outcome being from pure chance is 50%, which is extremely high. So we really can't say much about this. But again, there wasn't really many prescriptions of this particular drug. 7 for the poorly controlled group, and then 2 for the well-controlled group. So that really high p-value could just come down to not enough people participating in that particular variable. Okay, so the next thing I'm going to present is this graph, which I think really kind of encapsulates the result that controlled glucose is really important to good outcomes in COVID-19 and type 2 diabetes patients. So what this graph is kind of showing is the percent survival rates for the well-controlled cohort patients, as well as the poorly controlled cohort patients. So first, to kind of explain the graph, on the x-axis is given time in days. So from day 0 to a day 28. And then on the y-axis is given the percent survival, and these numbers are in percents. So it starts from 100% and it goes to 80%. So first, looking at the well-controlled cohort, we can see that over time, their survival rate kind of is pretty steady. It kind of hovers around about 98%, which is pretty comparable to people who actually don't have type 2 diabetes who also contract COVID-19. But then when we look at the outcomes for the poorly controlled cohort, we can see that over time, their survival rate actually dips down really dramatically compared to the well-controlled cohort. It dips down to below 95% to about 93% at day 28. And this is, I think, the most significant results of this study, because it really demonstrates that even though the poorly controlled cohort patients, from the last slide, we know that they had more medical interventions than did the well-controlled group. The well-controlled group is still surviving at a much higher rate than the poorly controlled group. So this kind of implies that one of the most important things in good outcomes for COVID patients with pre-existing type 2 diabetes really is just good control of blood glucose. And remember, healthy blood glucose in the study is defined between 3 and 10 millimolar per liter. Okay. So finally, I'm going to talk a bit about the conclusions in this study and the specific limitations, as well as future studies for this kind of research. So first, I'm going to talk a bit about the clinical significance of the result. So this is kind of a cartoon, actually from the paper itself, and I think it really presents the most important clinical outcome from this study really well. So it kind of depicts the previous graph in a kind of cartoonish way. So the blue line is the survival rates for the well-controlled blood glucose cohort group. And then the red line represents the survival outcomes for the poorly controlled blood glucose patients. So what this is kind of depicting is on the blue line is kind of a person who is like battling COVID kind of really well, and they have a survival rate of 98.9%, which again is really comparable to patients, COVID patients who don't have type 2 diabetes. So, and this I think really demonstrates the importance of maintaining good blood glucose when diagnosed with COVID-19. And then in comparison, on the red poor outcome line is a person who's depicted as kind of like falling as a result of COVID-19. And these individuals have a death rate of about 11%, which is really, really a dramatic decrease from the 98.9 survival rate of those who have well-controlled blood glucose. So again, the clinical significance here is that really one of the most important things in these individuals who have COVID-19 as well as pre-existing type 2 diabetes is really strict control of their blood glucose. Okay, so now I'm going to go a bit into the limitations of this study as well as some future directions of this research. So first, a really good way that this research could be extended is through the performance of a similar, what's called a prospective study, in order to supplement the current study, which is, again, a retrospective one. And what a prospective study is, it's really similar to retrospective studies, except that when you're comparing your two cohorts that have some variable that you're interested in, what you're doing is you are kind of tracking them, tracking the outcomes, excuse me, in real time, rather than working with data that was previously collected as in a retrospective study. Okay, another thing that could be done is to increase the type 2 diabetes cohort size. And if you remember, the total cohort size was approximately 7,000 patients, and then the type 2 diabetes cohort size was about 900, which is really, really small, obviously, compared to the total cohort size. And size of the cohort really matters in these kinds of analyses, because the statistics that the researchers take advantage of in this case really depend heavily on size. And when comparative size, as in this case, is really small, we can get a lot of what's called confounding factors in our analyses, which are basically just factors that we don't necessarily care about, but are affecting our results. Okay, so another thing that we could do is to understand the comorbidity between COVID-19 and type 2 diabetes at a more fundamental level. And what I mean by fundamental is more of at a kind of basic science level. So understanding the comorbidity at like a biochemical level, at a biophysical level, so that we could potentially create a really specified specific treatments for individuals who suffer from both type 2 diabetes and COVID-19. And this could manifest in pharmaceutical treatment, but it could definitely be something else. Okay, and the last thing is to make this study, to extend the results, is to make this study more multi-centered, and that is to perform it in more diverse communities. And as I had mentioned previously, both COVID-19 as well as type 2 diabetes affect certain populations really at a disproportionate rate. And if you remember, the current study that we're talking about now was done only in Hubei, in the Hubei province in China. So performing a really similar study to this one would really help in kind of getting better health outcomes for particular communities that are affected disproportionately by both diseases, because these communities could potentially have really unique factors that affect them, which don't affect other communities. So again, just making it more multi-centered would really help to impact the positive health outcomes that could possibly come out of this kind of study. And kind of just to further that point, I'm going to present some data about diabetes diagnoses in the United States as delineated by race and ethnicity. So if you look on the last three bars on this bar graph here, you can see that Hispanic people, Black people, as well as Native American people are diagnosed with diabetes the most frequently out of any other race or ethnicity in the United States. And then this data kind of presents something similar, but with COVID-19. So it presents the percentage of deaths due to COVID-19 as delineated again by race and ethnicity. So from this data, we can see that Black and African American people, as well as Hispanic and Latino people, make up 14% as well as 19% respectively. And this is data from New York State. So this is their populations in New York State. But they actually make up more than 50% of all total COVID deaths, despite making up much less than 50% of the total population. So just taking this data, as well as the data that I presented in the last slide, and kind of going back to why this study is really important, because obviously this is extremely disproportionate, and again, both COVID-19 and type 2 diabetes affect these communities of color really disproportionately. So this kind of study that kind of looks into the interactions and intersections of both of these diseases, which again affect these communities at disproportionate rates, is extremely important to developing really positive health outcomes, as well as just providing better medicine to these communities. And again, going back to my thing about making this study more multi-centered, again, these communities can really suffer from factors that are really unique to them, which create these disproportionalities. So making this kind of study more multi-centered and more focused on these communities specifically can really impact these communities in a positive way, and again, create really amazing health outcomes, as well as just improved medical treatment of these communities in general.